Hello everyone and welcome to our next video. Now, in this video, we'll be discussing the basics of what is cancer, the hallmarks of cancer, and very few basic things which are essential for anyone who was introduced into the field of cancer. This is considering the first year MBBS students especially who might be new to the pathological concepts of cancer as well. So without further ado, let us start. What is cancer? Cancer is a group of diseases characterized by cell growth, invasion, and spread. So there will be a primary site. From there, it can remain constricted to that place, restricted to that place, or it will metastasize to other places as well, invade through the primary site to other body tissues. Now, the hallmarks of cancer, there are 8 hallmarks. Growth signal autonomy. As we all know, normal cells need external signals from growth factors to divide. But then cancer cells, they are independent of this. This is because acquired mutations short circuit growth factor pathways leading to unregulated growth. Next is the evasion of growth inhibitory signals. Now, cells don't grow just like that anywhere and everywhere in the body. That's because of growth inhibitory signals which keep this in check. That is, they don't allow cells to divide continuously just like that. But in cancer cells, they do not respond to growth inhibitory cells, and that causes a problem of continuous division. They avoid immune destruction. Now, our body can recognize and eliminate virulent cells or cells which are harmful to us as such. But cancer cells do not stimulate an immune response, and they can interfere with the immune response so as to avoid immune destruction of these cells. But uh, like emerging therapy modalities involve making our immune system recognize these cells by using vaccines or so and so, and then making our immune system to destroy the cancer cells. I had covered such sort of treatment modalities in our previous videos. So next is unlimited replicative potential. These cells, the normal cells, become senescent because of shortening of chromosomal lengths, telomeres. Now, cancer cells maintain the length of their telomeres, and this results in unlimited replication potential. The next thing is invasion metastasis. Normal cells maintain their location, do not go here and there as and when required. But as far as cancer cells are concerned, they can uh, move to the other parts of the body as well, and they can invade the local tissues too. The next feature is angiogenesis. Now, this is the ability of cancer cells to induce new vessel formation, new blood vessels that help in tumor survival and expansion. Genome instability is the other feature, which is like um, there will be faulty DNA repair pathways that contribute to genome instability, and this is like basically alterations or mutations causing in the genome of cells causing this unlimited replicative potential. And we might know about apoptosis and the whole mechanism as such. I'm not going any further into that, but the cancer cells evade this programmed cell death and they just live like that. Now, let us go to the next concept, oncogenes and tumor suppressor genes. There are oncogenes and tumor suppressor genes. So what are these? Basically, a proto-oncogene is like a um what do you say? Like pre maturing or premature form of an oncogene, which is not activated yet. It can get activated into an oncogene by mutation as such. Or, in other words, oncogene is like a cancer causing gene. Now we have tumor suppressor genes which inhibit this process. As in, there are gateways of what do you say in the cell cycle which inhibit the tumor growth as such. We'll get further into that in the upcoming part. Now, factors influencing carcinogenesis. These involve smoking, alcohol, diet, and environment. General factors. They Each of these might cause mutations or um, difference in cell characteristics that may produce cancer. Now, the basic principle of chemotherapy. Conventional chemotherapy targets DNA, RNA, and proteins to disrupt the cell cycle in rapidly dividing cancer cells. Now, the goal of this is to cause DNA damage and trigger apoptosis in cancer cells. They affect healthy, fast dividing cells, such as hair follicles, stomach lining, blood cells. Each of these has subsequent consequences like hair fall. As the hair follicles are affected, then ulcers might be there as the stomach lining is affected and blood cells getting affected cause anemia. Now, toxicity is the main feature of this. If we look at some of the chemotherapeutic agents, the therapeutic index, that is like the dose range within which it should be maintained to cause toxicity, it's like a very small range. That is, we need to constantly maintain it within a certain plasma titer level, so that it doesn't cause toxicity as such. Now, coming to the molecular pathways in carcinogenesis, they are interconnected with some of uh, these four displayed on the screen acting as star players. Now, what are protein kinases? They are key in cancer biology as they add phosphate groups to specific amino acids in proteins. There are several types like tyrosine kinases, which phosphorylate tyrosine residues, serine or threonine kinases, which phosphorylate serine or threonine residues. Their functions include cell cycle progression, signal transduction, and transcription, and they can be found as transmembrane receptors, intracellular transducers, and nucleoproteins. They're targeted in cancer drug design. Now, they are regulated by phosphatases, which remove phosphate groups, and some act as tumor suppressors. What is the next thing? So we are done with protein kinase. RAS family. They're mutated in over 50% of certain cancers, such as in colon cancer, and they function as intracellular transducers transmitting signal from growth factor receptors. They reside on the intracellular side of the plasma membrane and are activated by GDP-GTP exchange. That's all. If at all anyone requires the further explanation of how RAS comes into play, or any of these come into play specifically, please mail us. Our email ID is available on the channel, so we'll be getting back to you personally. P53. The next one. This is the guardian of the genome for its role in cell cycle arrest, DNA repair, and apoptosis in response to stress. It acts as a transcription factor, inducing gene expression to inhibit carcinogenesis. It is mutated in over half of all the cancers with thousands of identified mutations. Next is the retinoblastoma gene. This is a tumor suppressor that regulates the cell cycle, and it is mutated in various cancers, especially retinoblastoma itself. Normally, it inhibits cell cycle proliferation by suppressing a transcription factor essential for cell cycle progression. Now, this is regulated through phosphorylation by cyclin D and cyclin-dependent kinases. I'm not further going into specifics of how these are regulated in turn with respect to cell cycle, but just know that this is a tumor suppressor gene as such. Now, if we come to retinoblastoma, there might be familial mutations. Suppose there are like two, um, what do you say? Yeah, two, you have a gene, you have two alleles. One of them is already mutated in the familial pattern and the other is free. Now, this increases the chance of both of them getting mutated, and that is basically Knutson's two-hit hypothesis. So, if one more gets mutated in the future, that will lead to development of cancer as such. But in sporadic cases, both of them should be mutated together, and that is relatively rare as compared to familial. So that's all for now. Thank you, everyone.